Hello, everyone. Thanks for tuning in to the second chapter in our series of muscular system lecture. Today, we're going to cover the microscopic anatomy of the muscle tissue. And remember, we're talking about skeletal muscle tissue now. Okay, so if you remember from the last lecture, we talked about the gross anatomy, and here you see the gross anatomy of a muscle. We talked about the different coverings of connective tissue and um, how it's kind of built like a rope. It's bundled into different fibers. We have the bundles called the fascicles, and then each fascicle has lots of muscle fibers. And remember, we call muscle cells muscle fibers because they're much longer than they are wide. So they kind of look like a thread, like a fiber. So today, what we're going to do is we're going to go down to the cellular level, and we're just going to look at the muscle fiber, and it's going to be here where you're going to see some of um, those fibers and proteins and macromolecules that allow a muscle to contract. We're not going to get into contraction yet, but we're going to look at the different components of contraction. And so we're going to look at this part right here. We're going to look at the muscle fiber. So here's our muscle fiber, and you can see that inside the muscle fiber there's even more. They look like fibers, but they're called myofibrils. But let's look at the muscle fiber, and we know that the muscle fiber is called the sarcolemma. And so the sarcolemma, remember, is the membrane. It's kind of the cell membrane of the muscle. And what's kind of unique about um, the cell membrane, the sarcolemma, is that it's kind of transparent, and you can see all of those nuclei right under the surface of that sarcolemma. Remember that skeletal muscle fibers are multinucleated, so we can see all of these nuclei right under the surface, and that's because they're pushed out. The muscle cell is so full of these myofibrils that there's no real room for anything else, right? So you also see on this diagram, we have these tiny structures here that don't look like or that don't look like myofibrils. These are the mitochondria. Skeletal muscle cells have one of the highest ratios of mitochondria um, to cells in our body because our muscles need a lot of energy, and of course, mitochondria make ATP. So let's look at what this myofibril is. It must be important because our skeletal muscle is full of them. Um, that's one of the main components of the muscle fiber. And notice we're using those um, prefixes, right? Myofibril. Myo means muscle. Sarco, right? Sarco means flesh, like sarcophagus. But remember to look for those prefixes. So if you think about a muscle, a skeletal muscle, and when you think about what it's composed of, you can think about what you eat for dinner if you're a meat eater, right? Because we say, if you want to get protein in your diet, the best way is to eat lean meat. And so it should be no surprise that this muscle fiber, your muscle cells, are full of protein um, and protein molecules. So this myofibril is one of those protein fibers. and it's full of its own protein molecules, and we'll get to those in a minute. So we have the muscle fiber here. The outside of the muscle fiber is the sarcolemma, serving as a cell membrane. You can see those nuclei scattered around there, pushed out to the outer parts of the muscle fiber, the mitochondria, and then this myofibril. Over here it talks about the dark band and the light band. We're going to get to that in a minute, but note that the dark band is also referred to as the A band, and the light band is referred to as the I band. And I always remember that the light band is the I band because the light has an I in it. So anyway, moving on. So again, talking about the sarcoplasmic or the sarcolemma, that's the plasma membrane. Your book is just kind of emphasizing that prefix. And then we also have another structure called the sarcoplasmic reticulum. The sarcoplasmic reticulum is a kind of specialized smooth endoplasmic reticulum. And it's the sarcoplasmic reticulum that stores calcium. So if you want to write that in there, it stores calcium, and this is really important for when we start talking about muscle contraction, because calcium is the final trigger for muscle contraction. So here we have one myofibril. And so let's take a look. Now we're down to the molecular level, right? So we've left the cellular level, we're down to the molecular level. We're going to talk about some of those. We're going to talk about some of those protein molecules that make up this myofibril. So the basic structural contractual unit of a myofibril is called a sarcomere. We'll get to the sarcomere in a minute, but a sarcomere goes from one Z disc to the next Z disc, and you can tell why they, call these Z discs. When they discovered these, when they first saw these myofibrils, they're looking at them under a light microscope, and they see, the zigzag lines here, and it looks like a Z, right? So that's our Z disc. And then the entire myofibril is composed of our myosin filaments and our actin filaments. And we need to know that the myosin filaments are the thick filaments, and the actin filaments are the thin filaments. And you can see within this sarcomere that we have an area where, I'm going to go back to red, where we have these myosin filaments here, right? And also, you can see where we have the actin filaments kind of overlapping, right? The overlapping of those filaments is what causes the banding, what gives us the light bands and the dark bands. So it's the arrangement of the fibers that gives us the banding. That's kind of important. You'll see that on a test. So it's where these bands overlap here is where we have our A band. You see that? That's our A band. And then out here, where they're only have what do we only have out here? We only have our you only see those thin actin filaments out here, right? And so that's where our I band is found. Okay, so our I band, there's no overlapping of filaments. The only filaments we have within the I band area are the actin filaments. And then here they're overlapping. Okay. List of words to protein molecules. Whoops, moving on. And like I said, the sarcomere is our contractile unit of the muscle fiber. And so what happens is we have the Z discs here, and it doesn't look like anything is attaching these myosin filaments here, right? This myosin filament, it looks like they're just kind of hanging out there. Well, they're actually attached here at the H zone, but they're also attached to the Z discs. And I did not put this on the agenda as one of the terms you need to know, but you do need to know the protein that attaches the myosin filaments to the Z disc, and it's called titin. I'll just write that. It's a very elastic. It's kind of like a spring. It's a very titin. It's a kind of a spring-like protein, so that those myosin filaments can move. But those myosin filaments are attached, and that's why I'm drawing them kind of squiggly, because they're kind of spring-like. They're very elastic. And let me try to write that just a little better. Since I didn't put it on your to know, it's Titin, titin. Okay, so you can kind of see how this contractile unit is set up. So what happens is that when our sarcoplasmic reticulum releases the calcium, and it releases the calcium in response to a change in the electrical voltage across our sarcolemma, but when it releases the calcium, there's a number of reactions that you'll be learning that causes the heads on these myosin filaments to attach to the myosin filaments and pull them towards the center of that sarcomere. So let's go look at it. So here's our sarcomere. Oh, here's a much better rendering of the titin molecule. Let me just circle that for you. So here's the titin molecule. So we have these thick myosin filaments, right? And the thin actin filaments. And you can see the myosin heads here. So I'm just going to make those a little bit more prominent. So what happens when the calcium is released, these myosin heads attach to the actin filament, and on their power stroke, they pull them towards the middle of that sarcomere. And what happens is that sarcomere then shortens. And you think, okay, so that's a shortening of one sarcomere, but if you look at how many myofibrils were packed into one muscle fiber, and think about all of the sarcomeres along each myofibril shortening, that's what gives you the shortening of the muscle and ultimately the muscle contraction. Okay, so we should know the sarcomere. We should know the Z disc, actin filament, myosin filament, and titin. Now, it says up here that the myosin protein has ATPase enzymes, ATPase enzymes are going to break down ATP to what? ADP, right? And they're going to release energy. And this is what powers our muscle contraction. This is what allows for each power stroke that those myosin heads pull that actin filament towards the center of the sarcomere. It takes some ATP. Okay, so it's the ATPase enzymes that that the myosin contains that allows the myosin to break down ATP to ADP, releasing energy that it will use to shorten the sarcomere. We pretty much talked about the organization of the sarcomere. Let's move on to the next slide. Okay, this is great. This is a close-up of what those myosin heads look like. And in your textbook, it calls them myosin cross bridges. I usually just refer to them as myosin heads. But like I said, once that ATPase enzyme breaks down ATP to ADP, it releases energy, and these heads are going to all attach. They're going to attach here, and they're kind of cocked back. And then when that ATP attaches, they pull it towards the middle, right? And you can see how that would shorten that that sarcomere and ultimately shorten the entire myofibril, which is going to shorten the muscle fiber, which is going to shorten the muscle tissue. And like I said, and we're gonna sorry, we're gonna talk about the sarcoplasmic reticulum in the next lecture series or the lecture installment. But I do want to note that the sarcoplasmic reticulum stores calcium, and it's calcium that allows those myosin heads to attach. Because you might read about it this week. There's blocking mechanisms on the thick filament. So there are where these myosin heads need to attach, the attachment sites are blocked. They're blocked by two proteins, troponin. It's called the troponin tropomyosin complex. So what happens is that the calcium molecule binds and it removes that troponin and tropomyosin, and that allows these heads to attach to the site. And like I said, once it attaches, it's going to pull it towards the center for shortening. Now, there's lots of steps in muscle contraction. Your reading for this week over muscle contraction, actually, yeah, you do read about muscle contraction this week. So there's a lot of information, and you would not believe the amount of steps that have to be there's so many steps to go through just to contract your muscles. So what I've put in the agenda is I've put a list of steps so you can kind of get an idea of the steps that happen in muscle contraction. There are also some great videos and animations for the generation of the action potential, moving down to the muscle, the sarcolemma, the neurotransmitter crossing the synapse and binding to its receptor on the sarcolemma and causing the action potential on that muscle fiber. So anyway, it's a good idea to look at those videos. And like I said, those videos are listed all week on your agenda in the resources. So, alrighty. Well, remember that we have office hours on Tuesday, and I'd be happy to take any questions that you have at that time. Otherwise, stay home and stay healthy. Have a great week.